As we get into the discussion of the endocrine system, we are transitioning from the nervous system, which involves chemicals, ligands, being passed from a presynaptic neuron to a postsynaptic neuron. We're transitioning from that understanding to endocrine system, which is an endocrine cell which makes a hormone. A hormone is also a chemical messenger and sends that messenger to a target cell. So I'm wanting you to understand that this is a gray area. Lots of the ligands, the chemicals, the messengers, can cross over between one system and the other. So it's not so black and white. In fact, a lot of the terminology is the same that we talk about in the nervous system as it is in the endocrine system. So the most important thing about chapter 6 is the signal transduction pathway. I want you to understand that this is what we're talking about. I often call it STP. For the rest of the semester, we'll be talking about signal transduction pathways. And this, in a nutshell, is how a metabotropic receptor works. So let me give you that overview, and then hopefully you won't get bogged down in all the details. I don't really want you to know specific names of enzymes or specific names of second messengers, but I want you to know if you see one, what it is. So for an overview, this is, it's a weird shaped cell. This is a target cell. Now that is a general word, the target cell, for a cell that has the receptor. So it has the metabotropic receptor here, meaning it can respond to the chemical messenger. And the only chemical messengers that we're talking about for a signal transduction pathway are polar. It has to be a polar ligand, which is sometimes called the first messenger, to use the signal transduction pathway. Because, think about it, if this ligand chemical messenger is trying to tell this cell, the target cell, to do something, if it's nonpolar, it can just cross right through the cell membrane. But if it's polar, then the messenger can't get the message into the target cell without using this receptor. The receptor is key because it is a transmembrane protein. It can bind to the polar ligand out here, and then it becomes activated on the inside. Okay, so first this is going to bind to the receptor. And this receptor is a metabotropic. In other words, it is not the kind of receptor that is one package. It is not an ion channel. It is a metabotropic receptor. And this is the kind that works from the inside. So the ligand binds out here, and it can't cross, but the receptor can. And the receptor becomes activated. So I'm going to say activate the receptor. And this is key because an activated receptor can do all kinds of things from the inside, from the cytoplasmic side of the target cell. Now, one thing you need to keep in mind is an agonist looks like, acts like, smells like the actual ligand. So an agonist could also bind here to the receptor and activate the receptor. Now, when the receptor is activated on the inside, usually it's bound to a G protein. Most metabotropic receptors are bound or attached on the inside, cytoplasmic side, to a G protein. So usually activation of the receptor will lead to activation of the G protein. The G protein discoverers were given the Nobel Prize in 1994 for the discovery of the G protein. That's how important it is. Not all metabotropic receptors are bound to a G protein, but most of them are. For example, if this is epi or norepi and it's binding to an adrenergic receptor, it is coupled to a G protein. If this is acetylcholine and it's a muscarinic cholinergic receptor, which is metabotropic, it's also bound to a G protein. So G protein is kind of like the middleman. And so when the G protein gets activated, then it can in turn activate other things. And the key here is that the G protein's job, there's lots of possibilities, I'm only going to give you two of them, is to either go over here and open or close ion channels. So that's why I said the channel is over there, and then it can open or close that ion channel. So that's one possibility. And the other possibility is to turn on or off enzymes. And so there's lots of specific details you could learn about this, like what's the name of the enzyme. But I'm not going to ask you adenylyl cyclase. I'm not going to ask you that. There's so many possibilities. When we get to them, you will know them. So for right now, I just want you to know the overview of a signal transduction pathway. And these proteins, like a channel or an enzyme, are existing. So I'm going to say change in existing proteins, such as enzymes and channels. Now, this is important because the protein is existing already inside the cell. The cell doesn't need to make enzymes from scratch. Using a signal transduction pathway, it can bind to the receptor. That's the signal first messenger. The receptor gets activated. Usually a G protein is the middleman. And then just alter the shape of an enzyme, which turns it on or turns it off. Or it can alter the shape of a channel, which opens it or closes it. This is relatively fast. Whereas if you had to make that protein from scratch, go into the DNA, make the messenger RNA, put the amino acids together, that would take forever. So this is relatively fast. And then maybe you open a calcium channel, calcium comes in, calcium could appear in the cytoplasm. It wasn't there before. Or your enzyme makes a new product that wasn't there before. If something appears in the cytoplasm, which wasn't there before, these are called second messengers. So your first messenger never comes in to the cell. It works through a signal transduction pathway. That's why the key to the first messenger in this pathway is it is usually almost always polar. There's a couple of exceptions. There's an estrogen receptor that does this, but most of them are polar. Non-polar things, like if it's a non-polar ligand, it just can cross right through the cell membrane, and it can find a receptor inside the cell. So it works not by a signal transduction pathway, but there's its receptor there, and we'll get to that when we get to the endocrine system. Okay, so this is the most important thing. The overview is you can very quickly use a signal transduction pathway to change existing proteins and cause a response in the target cell. That is obviously the end result here, is we're trying to get a response in the target cell. For example, when we talked about this with the autonomic nervous system, you could think about this being the heart, okay? And when the acetylcholine, for example, it binds to a muscarinic cholinergic receptor, we're talking about the parasympathetic nervous system. And it will activate this whole pathway, which is sometimes called a cascade or an amplification, or it could be called a second messenger pathway. And that will cause a response inside the heart cell to slow down the heart rate, to slow down the stroke volume. So that's the overall goal, is this pathway causes a response in the target cell. Please let me know if you have any questions. Thanks.